Let me just start with an overview. I'm going to talk about some definitions of genomics because I find oftentimes people don't really know what genomics means. I'll talk about genomic technologies versus genomic science. There's a difference between the two. And then I'll talk about genotyping and gene expression, which are, I think broadly speaking, the two major classes of functional genomic analyses that you can do and that people are doing. And lastly, I'm going to talk a little bit about a synthesis of the two ways in which you can use both genotyping and gene expression together in a branch of, rapidly emerging branch of genomics called genetical genomics, which I think is particularly interesting just myself and kind of shows you where things might go in the future. Okay. Before I talk about what genomics is, why don't I say what is genetics? And I know you're all probably familiar with it, but it's useful to kind of go through a list like this as a counterpoint. Pre-genomic technologies, as I've termed them, include traditional genetics and molecular genetics. Traditional genetics, going back to Mendel and other early geneticists, really didn't take much in the way of technology. It took good observational skills, some phenotype, meaning some trait that you could easily score and quantify in some way, and usually these were visual traits like roundness or smoothness of peas or wrinkled peas. The ability to carry out controlled matings, because genetics is all about inheritance of traits in offspring. That's the basic technology, if you want to call it that. Insights regarding breeding schemes. In other words, early geneticists realized that you could get a lot of information by doing back crosses, where you would cross offspring to their parents, and that would allow you to show the segregation of traits. And lastly, linkage analysis. And essentially all of genetics rests on the foundation and insight that traits co-segregate. And nowadays it's known that traits co-segregate because they're physically in proximity on chromosomes, and that large chunks of chromosomes are inherited in blocks. This insight early on allowed people to start to fabricate genetic maps where they said, well, any trait that co-segregates with another trait is associated with it, linked, literally, in the terminology. And this allowed people to kind of generate the first pictures of how hereditary units of information, which we today know as variations in DNA, segregate with the biological manifestation of the trait or the phenotype. It didn't take long for geneticists to realize that they needed more than experiments of nature, wrinkled peas versus smooth peas. They said we need organisms that work better than, say, peas, that breed faster, that are smaller. And genetic model organisms were devised, things like Drosophila and C. elegans, which is a nematode worm, fruit fly. These were seized on by geneticists because they could do things to them that made genetics 10 or 100 times more powerful than using, say, dogs or something that's got a long breeding scheme or that's difficult to control the breeding of. And these technologies include things like saturation mutagenesis, where they said, okay, let's take an organism and treat it with some chemical irradiation that's quite toxic, and that mutagenizes the organism to such an extent that you can score hundreds of phenotypes and everything you might think of. The eyes end up changing different colors. You end up having new limbs sprouting and so on. And this allows you to say, okay, any gene that leads to a sprouting of a new limb or a limb showing up somewhere else in the body, it must be involved in that trait. This led to other techniques I won't go into, like radiation hybrids, and ultimately culminated in things called physical maps. These are where people said, well, since we know that traits are co-segregating and are on single pieces of DNA, we can start to clone large chunks of DNA through genetic engineering technologies. And rather than using co-inheritance in offspring, we can look at traits that end up segregating together on chunks of chromosomes that we've taken out of the organism in molecular clones. And then, of course, this all culminated and was supplemented from the 70s through the 80s and 90s with sequencing, which became like the size and speed of computer chips, exponentially better with each passing year, to the point that in the late 1980s, people said, hey, why don't we just do away, or not really do away, but supersede all the earlier genetic technologies, and let's just sequence an organism. Let's just sequence the whole thing and generate that information. And the whole idea of genomics essentially was that the term was coined to describe those efforts to sequence the human genome. And of course, people didn't start with the human genome. They said, let's knock off some of these genetic model organisms first. So the early work started with something like C. elegans and Drosophila, yeast, and of course the human genome was finished in, I guess it was 2000 or maybe 2001. In any case, it's been done for 3-4 years, first in draft form and more recently finalized. When we talk about sequencing a genome, by the way, for most of these organisms, we're talking about sequencing an individual, a strain. In humans, I think it was about 20 individuals who were sequenced. There were two different, a public and a private effort, and they had slightly different strategies. But of course, when we say we sequenced the human genome, it's important to point out, and this is kind of the foundation of the talk, we sequenced a human genome, or maybe a few. What we didn't capture is what's out there in all of us, and that's what really kind of post-genomic science is about. So post-genomic technologies basically are all driven by the fact that this primary genomic information, which was generated by sequencing a few people, was there. Now the question ends up being, okay, it's not really a mystery as to where genes are. It's more a question, what do these things do? We've got this huge list of 30,000 genes. We only know the identity of fewer than half of them. And we don't know anything really about the variation. We have the Book of Life, but we have one edition, and everybody's edition is different. Everybody's got slightly different copies that somehow explain the differences between them, but we don't really know who's got what copy and which text gives which outcome, disease, trait, you name it. So that's really the foundation of the kind of disparity, or the genomic aspect of disparities work that is going on in our center and around the world. Okay, all of these terms have sprouted in the last 10 years. There's all sorts of different kinds of genomics, and don't let it confuse you. Really, all omics really is, and this is kind of funny, these are all terms that I got off the web from the omics glossary. People talk about the cell-ome, which is the entire complement of molecules and their interactions within a cell. The complex-ome, the degradome. In other words, pretty much people are applying this idea of omics, of a high throughput approach to everything. And in a way, this is kind of fatuous, and in a way, it's not. The way it's not fatuous is that people have finally understood that a high throughput discovery-based approach, where you just go in and you say, we're going to take technology, brute force, and try to measure a lot of things and then figure out what's going on later, is actually pretty fruitful. And so everybody's trying to jump on the bandwagon. So I think it's kind of funny, but I think it underlies the fact that biology is now entering a new era when high throughput approaches make sense. High throughput approaches, as I'll say at the end, have characteristics that are very different from the kind of genetic research and other kind of biological research that's gone on in the past. So omics is fundamentally associated with these things. Scale, we're talking about hundreds to thousands of genes or proteins or lipids, or hundreds to thousands of samples, oftentimes both hundreds to thousands of samples and hundreds to thousands of analyses of them. A philosophy that we are going to do discovery-based science. We don't start with a hypothesis necessarily. We just start with the general idea that if you measure a lot of things and you've designed your study correctly, you will come across interesting associations. The motivation, of course, is the following things: that we now have the whole genome sequence of humans and other species. We've got computing power, and even 10, 15 years ago, a laptop like this is extremely powerful, but these were expensive and difficult to get 15, 20 years ago. And automation, as far as what I do in the lab, which is kind of machine-based technology, the computers and robots have gotten cheaper and smaller and better, and they allow us to do high throughput work. Lastly, and this is the most important thing, is a perspective. Genomics is characterized by the idea that looking at a single gene at a time is anecdotal at best, and that you really need to go on from there. You can't just look at one gene, you have to look at interactions between genes. Also, this has been termed systems biology, which really can be summarized as complete knowledge. This idea that you will understand everything about an organism and have a quantitative, holistic, and predictive model for a disease state, a trait, and so on. And I'll get back to that at the end. Genomics is not the following. It's not the same as technology. A lot of the time people say, well, I want to do genomics, and then they say, I want to do genomics, I want to use that neat arrayer. And well, genomics isn't just the arrayer is just a tool. That's all. It's just technology. Genomics often depends upon the technology, and the practice of genomics is different. My lab looks very different from most labs. A lot of labs are kind of messy and artful, and a genomics lab has got to be extremely organized. And Esteban will tell you the worst thing you want to have in a lab is some kind of non-standardization, because when you're dealing with hundreds of plates that all look the same, you got to keep them straight.